So good morning, good afternoon, and good evening to the Pasteur Network community, and welcome to this new Pasteur Network monthly seminar. Thank you very much for being here. I'm Hisham, coordinator of these seminars. So, as always, we will kick things out with our speakers, followed by a Q&A session. And during that moment, feel free to drop your question in the chat or ask them out loud during the Q&A. A quick reminder to keep your microphone and camera off during the seminar to help everything run smoothly. Don't forget that the seminar is recorded and will be shared with our community on the Pasteur Network YouTube channel. Today's topic is multi-site management of biospecimen collection and associated data. And for that, we are very pleased to have with us our speakers, Laurent Jacotot, co-founder and CEO of Modul-Bio, and Mike Woodward, Business Development Manager in Modul-Bio. So Laurent Jacotot is a robotics engineer and the co-founder and CEO of Modul-Bio, a company specialized in software for biobanks and cohorts projects, with more than 25 years of experience in process automation and databases applied to biology. He has worked in Boston on the Human Genome Project at the Center of Genome Research Whitehead Institute and at the Dana-Farber Cancer Institute, and finally at the Genomics Collaborative, a private biobank. Mike Woodward has an experience, is very experienced in highly skilled business development manager and consultant for specialized biological software products, with over 20 years of experience working in the public and private sectors. Originally, he is a forensic scientist, then worked internationally to provide and train interpretation software before moving into biobanking. Mike has proven ability to cultivate, build, and maintain principal relationships with customers and stakeholders. He works with clients worldwide to enhance their biobanks and projects with mBioLIMS biobanking software, and has the ability to drive positive technology change. Okay, then. So now I'm happy to leave the floor to our speakers, and I will begin with Mike Woodward. So Mike, if you can share your screen, that would be great.
Yes, of course.
Thank you very much.
Hopefully everybody can see the screen now.
Yeah, we can see it. Thank you.
Excellent. So yeah, thank you for the introductions. Hello to everyone, wherever you are in the world, and also a big thank you for having us here today. It's a great opportunity to talk about what we do. So thank you for the time, and as stated, if you've got questions, we'll be very happy to go through them. So we're going to do a presentation on multi-site management of biospecimen collections. So we're going to look at different scenarios, different clients that we work with. And I'm going to talk in a little bit of detail about the projects and how mBioLIMS biobanking software is used. We may have a little time at the end to get into the detail of the software, but we can certainly talk about it offline as well. So just a little about Modul-Bio. We've been around for over 20 years building a biobanking software. And we specialize in biological sample collection. So that could be human or it could be non-human samples. And we've provided this software, and you'll see this as we go through the different examples, to different types of biobanks, cohort projects, biorepositories, some clinical trials. And you'll see that all of these projects are in the same area of biobanking and biorepositories. We don't build a generic LIMS. It's a very specific product for a very specific market. LIMS for biobanks, if you will, but we like to call it BIMS. So we do this with a team of different people at the company. As you can imagine, we have quite a few software engineers and experts, but we also have a mixture of those from the life sciences, including myself. And what that allows us to do is to work with the software engineers with the experience of lab work, understanding the protocols and the processes that go on to continue to build the project, to do the product to do what people need it to do. And we also take on board, and we've built the software doing this, the experiences of our existing clients and the functionalities that they need, or changes in the biobanking industry, and we produce new functionality and new modules to support that. So currently we have over 300 laboratories using the solutions, and we'll look at a few of them in detail. And the headquarters of Modul-Bio is based in Marseille, France. We have an office in England where I'm based, and we also set up a subsidiary in Canada. And what that allows us to do is to have kind of 2 time zone support for the majority of our clients. So most European clients are supported by our team in Marseille, whereas most of our North American clients are supported directly from Canada. So we're going to concentrate mainly on mBiolims and the projects that it's involved in, but we'll just go through the different things that we have done. So mBiolims biobanking software is a modular software we've built for these samples and associated data. We also use that as a built-in API for a plethora of different things, one of which could be a web catalog where samples are promoted and researchers can view and request those samples. And also built into mBiolims is mBiolabel. So it's an integrated barcode solution. Unique identifiers can be generated and barcodes can be scanned directly into mBiolims as well. So we'll concentrate mainly on mBiolims, but we certainly can talk about some of the other topics. So as I said, it's been designed for biobanks. Its interface, its functionality is built for those purposes. You can see just a snapshot of the software there. We've got an initial dashboard which gives you an overview of everything that's going on within the biobank or biobanks. And on the right hand side, we've got a working list, which I'll talk about briefly. So it's a very intuitive interface, very easy to use, very easy to administer, so you can create new things in your bank, new sample types, new studies, new storage containers. And we've also built in this working list you see on the right hand side, which kind of serves 2 purposes. One is a shortcut to records, so shortcuts to sample records or shortcuts to a patient record or a box of samples, but it's also a way to push or drag those samples into processes, so into storage or into a DNA transformation process. We've also added a messaging system which allows everybody to collaborate with each other. You can send messages to colleagues, send requests to colleagues, and a powerful query system's been built in as well. So you can interrogate that data. So a good example of that would be you can build a query looking for samples with specific metadata, locate those samples, drop them into the working list, send a message to the researcher or a colleague who was interested in those, ask them to aliquot the samples, and then ship them to the person who's actually requested them. So all of those tools kind of link together to have a really user-friendly way to manage the samples. And we've built it as a modular system, and we continue to add modules and functionalities to it to support this process within a biobank, of starting with a patient getting consent and samples directly from a patient and a specimen collection, then working with those samples, whatever processes they may be, aliquoting or assays or a transformation from blood to DNA. And we follow that journey all the way through their storage history, any further data that's added to those samples, any shipments or movement of those samples, to the end goal really of reaching researchers and helping to provide new therapies in precision medicine. So we've dedicated the functionality to the management of the samples and the data, because the data really is key in that process when you get to the end. It's evolving all the time. We've actually got a new version coming out relatively soon with some new features. And we've also designed it so it can be personalized. So rather than having to ask us to change things in the system all the time, there's ways for system administrators, client administrators, to personalize it, to create different user groups, to add new items when needed without having to come back to us. So it's a really intuitive and collaborative way to work, not only with the data, but with colleagues as well. And I think some of the examples we'll look at will show you that as well. So just a quick note on the modules. We may have time to dig into the detail on some of these later on, but they're really all the core needs of a biobank, the storage of the samples, the collection and the consent management, the patient management, which could be anonymous, it could be nominative data is hidden, and then the connections to different things that may exist in our clients' lab. It could be analysis machines, it could be 2D rack readers. They maybe want to track the temperature of the storage. They might want to connect to a hospital database to get their patient information. So we've built little tools and modules to facilitate all of those different things. And again, from a data point of view, there's reporting. There's a reporting module that enhances what you can do in the general search and the query builder as well. Okay, so we're going to start to think about the different use cases and concentrate really on some multi-site scenarios as well. And I just want to draw your attention to an article we wrote in Biopreservation and Biobanking, because that was kind of the topic of that article as well. And we looked at different scenarios, listed them, talked about some of the challenges and the successes on each of those projects. So we can certainly send a link if anyone's interested in finding that article. And in that, we kind of grew in size. So we thought about many of our clients are single biobanks, quite small teams, have a collecting center within a hospital. Then we looked at research institutes and academic center biobanks. And then we started to look at multi-teams within the same institution, and some of our diagnostic clients are good examples of that. And then we thought about national cohorts, clinical trials, and then some of these campus-wide solutions that some of the campus-wide solutions that I'm going to talk about as well.
We can't hear you, Mike.
Because he's been muted. I think he has been muted.
Oh. What's happening?
Oops.
How do we change it? Oh no, do you have an idea?
No, because okay, no, that's okay.
He's back. He's back.
I'm back. Okay, so what are the advantages of this concept of one software across multiple sites and teams? We're going to look at those, but the advantages are quite numerous. So the first thing is standardization. Standardization of processes, nomenclature across different teams, across different sites, means you've got a bigger data pool to search across. And a really simple example of that, and this was from back in my forensic time, was and much older databases, was the naming of samples. So if a sample was supposed to be DNA or RNA, if someone added a dot or they put a dash in between, it immediately segregates that data. When you're doing a search, you have to do a multi-line search to find all of those samples. So this allows everybody to use the same nomenclature, the same processes, the same standard operating procedures, and it just makes the data much more valuable. And also the concept of having unique barcode identifiers for samples and patients. The system won't allow a duplicate identifier, so that again is a way to standardize and also reduce errors. The more users, the more sites that are involved, allows them to collaborate, to share, and you can share samples, you can share working lists, you can share queries, you can send messages that the system saves. So again, there's a build-up and an audit of that collaboration that happens. One of the examples we'll look at as a specific example in the next few slides is about looking at data across multiple sites. So rather than having two different databases, two different approaches, you can query directly across all or 2 sites or whatever the search needed. And that shows you, as a very simple example, the availability of samples that are on a different site to yours. The requesting of samples that we've built into the system again becomes more powerful when you can do that, and you can also track the shipments between the sites. And think about this as real time as well, because as soon as a sample enters one site system, everybody who has access to view that can then see that. Same for processes. So a new aliquot or a new process has occurred that's produced some new derivatives. Again, this is all real time, so you can see it in the system. So on top of that, we then built some options to allow collections to be shared with other sites, or even lock down the visibility of certain sites to what they can see across the whole infrastructure. And we've got some experience with the Pasteur Institute already with this approach. Okay, so here's the simplest example really of the concept of sites in a single biobank. So the big Modul-Bio label is the biobank, and they're all different departments within one hospital that have embryolims but are collection center sites. So their functionality will be less than the main site. They can collect samples, they can add patient details or consent, and then they can ship them to the main bank. But the main bank will know that those samples have been collected because they have visibility of all of those collection sites. They can track the shipment and then scan the samples when they get them to ensure that the right samples are there. And there's a few other nuances that come into play here that are pretty useful, like non-compliances in the system or issues trackers. So you can check that the samples are in the correct order, they've been stored properly, or anything else that may have gone wrong with the collection or the shipment process. Now we're going to look at a slightly bigger scenario of this, again with one bank, but multiple collection centers. And this is across different countries in this scenario. So collections are happening at different places around Europe. Again, they're then shipping those to the main bank. The main bank already knows they've been collected because they're in the same software. And this is an example of it with the largest HIV biobank in Europe. It's growing all of the time. Different sites are being added, different collection centers. And at this point, they have over 180 sites connected in one system across many countries. It's around or over 40 countries at the moment. Over 1,000 users using this system, either at the main bank or in the on the main trial or in the collecting centers. And the number.
Attends, je baisse la réunion. Tu dis que tu es chez le.
Go ahead.
Yeah, each
Mike.
time we were having this problem, we were losing Mike. Mike, you're muted again. And
You're muted,
I am muting the people that did not mute their own microphone. So Sarah and Hiba before, but I did not do that for Mike. So that's weird.
Okay.
Okay, I've just unmuted myself here. That seems to have worked again.
Thank you very much.
So yeah, I'm not sure when I was muted. I'll just go over this again. So this is a growing project over time. The collecting sites has grown to over 180, over 40 countries now, over 1,000 users. So it's a pretty big connected web of different collections that are in these trials, and even the trials now have grown to over 190. So 2.1 million samples or thereabout in the system, and imagine the ability to look across and query and review all of the samples and the data that's been collected. This is a slightly different example. This is a national project, and you can see on the diagram we've still got collecting centers, but what we've actually got now is interconnected biobanks. They're different biobanks connected in the same system. So to us, the term site just means a different instance, a different team or a different collecting center or even a different biobank, and this can be managed and created within the software. So in this case, it could be they're all connected and they all share data, or they could even be siloed banks that just use the same software. They don't share anything with the other banks. They just work in the same manner in the same software. And an example of that is a client we brought on board a few years ago. Was basically the national Lithuanian biobank cluster, and there's currently 6 biobanks across the country that are all connected. They all use different collecting centers, and there's one central biobank. So 5 of them work in silos on their own and ship sometimes to the central biobank, and the central biobank has visibility of all of them, so they can query across all of the data of all of the biobanks in this national cluster. And I think for clarity, I think 2 of them actually do share data as well, but you can silo them completely, or you can share certain studies or trials as well. And interestingly, I was then contacted by another biobank in Lithuania, and they decided to remain siloed and not connected to the cluster. So they have their own instance of embiolyms. Although I think that will change over time, because there's communication about they still send information to the main cluster, but they're not on the same embiolyms instance. I think that will change over time. And then we're going to look at some of the projects we have in the US. So again, we looked at a different way of working in how they do this at campus level. So again, we've got various banks connected here, various collection centers within the university campus as well, and some outside. And this has been quite an interesting model to work with, because we introduced it when we first introduced it. The first one was UCSF. We trained a team at UCSF that would be the embiolyms experts, if you will, on campus at UCSF. And they were the first bank to come on board. They have an IT team structure behind them as well. So they know the ins and outs of embiolyms. They can help other banks. They can train other banks how to use them. And what they've been doing is adding a biobank at a time. So the next one comes along that wants to join the group. It could be a completely siloed bank, but they want to be on the same system. They're introduced, their storage is set up, their legacy data is imported, and they join the campus-wide solution. And we have got some discussions going on with one of our American clients that work with other studies, sometimes very short studies around the world. So in these cases, they can look to add those studies in temporarily if necessary. Again, they'll be able to visualize the data from America, maybe ship to the campus, but it's another use of this concept of a site in the software. So we've currently got 3 US universities using this model. We haven't done this type of model in another country yet. It hasn't seemed to have matched some of the big universities we work with elsewhere, but it is a very interesting and successful way to work with multiple banks and multiple collecting centers. As I said, we maybe get into the detail of modules later. I'm just going to hand over to Modul-Bio CEO Laurent to go through a few more slides, and I'm certainly going to be around for any questions later on. So again, thank you for your time.
Okay,
thank you very much, Mike. I give the floor to Laurent, who is our second speaker.
Thank you. Are you seeing slide 17?
Yes, we do. Yes, we do.
Okay, perfect.
Thanks.
So, hello everyone, and first of all, I would like to thank you very much, Isham, for the organization of this webinar, and I will continue to give you more detail about the MBioLIMS biobanking software. So, this software has been built to provide a centralization solution to allow users to work collaboratively in real time. It is installed on a single server. It can be installed on-premise, but we've done more and more installation in the cloud, and we work with the Oracle cloud infrastructures that provide health data hosting. So it's a very secure way of hosting the software, as everything is fully encrypted. We can do, it is very simple to manage and to deploy and maintain a multi-site application, as we have only to work on this server. We provide perpetual license, either user-based or as an enterprise licensing, which provide license for an unlimited number of users. There is no need to install anything on any computer. You just need a web browser like Google Chrome or Firefox. It's respect, confidentiality with a really good data encryption at rest, meaning when the data is stored inside of the database and as well as in transit, we use HTTPS protocol. And the system is also compatible with VPN, virtual private network, if you need even additional security. Very important too is that this system will integrate with your QMS certification project or any regulatory aspect, as it complies with the biobanking norm, the 2387. It complies also with GDPR, FDA 21 CFR, OECD guideline, good laboratory practice, HIPAA. There is a full audit trail inside of the software that allows to track each action of each user, what was the previous value, what is the new value, when it got changed, and for which reason. So the benefits really that we can provide maybe through some kind of a partnership is to provide really our experience. As Mike showed on the previous slide, I think it really explained how much experience and many different kind of projects that we've seen. We've done a numerous one, and we gain a lot of experience and expertise in the terms of the biobanking. It's really a software that has been built from scratch. We are not using a generic LIMS or any other types of software. We really built it from scratch. So it's really designed for biobanks. So it's very efficient, very intuitive. You don't have to do a lot of clicks to record aliquots. It is very simple to use, as well as very robust, because we use very good software technology, and you can have many users, many samples inside of this software. With, for example, what we've done with the largest HIV biobank that is located in Bordeaux, and that can really help to manage complete life cycle of biospecimen for biobanks, cohorts, or clinical trial. And we've recently added a new feature that will be released really soon, maybe even next week, I think. It's the ability to manage both human and non-human strains, within the same software, in the same application. In terms of fields where we work with many different biobanks that works on different pathologies. Of course, we work on cancer research, but not only. We work on diabetes, we work on virology, and of course, infectiology. And just to, you will see a few names here that I'm sure you know. On diabetes, we've done things for in the Emirates, for New York University in Abu Dhabi, as well as for the Imperial College Center for Diabetes, where they work with some clinics around Abu Dhabi to collect, biospecimen from patients inside of these different clinics. And there is a transporter that is going through the different clinics to gather the sample and bring them to NYU biobanks. So again, this is a different way of having biobanking. You could have a biobank that see a patient and perform sample collection, and you have biobanks that don't see any patient, but that works with collecting centers to collect samples from patients. We work also, of course, with the Pasteur Institute in infectiology, with the ICAREB, and we are deploying now another LIMS for the strain bank at Pasteur Institute Paris. And on virology, we work with the level 4, so very highly toxic virus with the P4 laboratory that is based in Lyon, in central of France, as well as with the French Army, the DGA, that has also a lot of viruses such as Ebola and some others that we don't know the name of, and the other one that I mentioned on the HIV. We've done very large cohorts, I would say, just to illustrate maybe this one. The ELF cohort is a very ambitious project where the goal is to collect samples from a newborn and from their mother, so 20,000 mothers and 20,000 children over the course of 20 years. So this is a very, very large cohort, and that involves many different hospital, maternity, and then some biobanking storage sites that are, I think, around 10 dispatched in the French territory. We work also on a big national cohort on breast cancer with 20,000 patients, and some others, across 23 hospitals for cryo-LIA or cryo-STEM project that works on, this one is on leukemia, on kids and teenagers, and cryo-STEM that works on graft versus host disease, that is also deployed at the whole national level with 25 different biobanking units. Just a few on them, but Mike already mentioned with Duke, Stanford, University of New Mexico, UCSF. This one is on it's on RareNet. It's mostly focusing on lupus with some different 11 French hospitals and 9 German hospitals that works collaboratively together on this disease. An interesting thing is that they exchange every 6 months or every other year their collection to create backups between those main sites, either the French and the German centralized repository. We work also, of course, in the private sector with some biotech diagnostics and cosmetics company that need samples to manage either to test, perfume or different chemicals for Christian Dior, or that works on new therapies or new diagnostics mechanism. That could be on colon cancer or lung cancer, for example. This is my last slide before maybe we move on to some questions, if you wish. We could propose, to work, and that's really something that we do with all with the biobanks and cohorts that we provide the software to, is really to work collaboratively, as a partnership. And we really specialize in biobanking and cohorts. We don't sell any other types of software. We're a member and founder of the ESBB network, and we're also a member of ISBER. And we try to provide really a personalized and scalable solution. And the Modul-Bio team is providing, professional services to accompany the software, such as the installation, the project management, the training, even the data migration for migrating your existing data that you may have into your biobanks right now that may need to be transferred to a new software. So we do all of that, and this is also, of course, supported with a dedicated project manager. Okay, thank you very much.